Okay, we're going to start our second lecture in cardiovascular physiology and talk specifically about the heart. Okay, so cardiac physiology is what is happening in your heart. So we're going to do a quick overview of the heart and do some basic anatomy. So if you haven't taken anatomy, everyone will be on the same page. Then we're going to talk about the electrical activity of the heart and get excited. Because if you like action potentials, the heart fires two different kinds of action potentials that differ from neurons and skeletal muscles. Okay, so we're going to talk about two different types of action potentials that the heart has. Okay, and then we'll talk about the cardiac cycle, and the last thing we'll talk about is the control of cardiac output. So this is all in chapter 13. Okay, so we're talking about the circulatory system. We already talked about blood, so that's the transport medium, okay, that's carrying all the oxygen and blood cells, etc. Okay, what we're going to do the rest of this week is talk about the heart, which is the pump. Okay, in order to be a pump, the heart has to be both the lowest pressure point in the vascular tree and the highest. Okay, so in order to pump blood effectively, your heart has to be the lowest pressure point at times and the highest pressure point at times. Okay, so it establishes the pressure gradient because blood is a fluid and fluids move down pressure gradients. They're going to move from where they're in higher pressure to lower pressure. And then when we come back from spring break, we're going to talk about the blood vessels. Okay, so your heart pumps your blood through the blood vessels. Okay, so let's talk about some anatomy of the heart. We have four chambered hearts. We have two atriums. So here's the right atrium, here's the left atrium. And we have two ventricles, the right ventricle and the left ventricle. Okay, so we have four chambers. The right side of your heart is receiving deoxygenated blood from the systemic circuit, so from your body. Okay, so this is why the superior vena cava is being color-coded blue, because this is deoxygenated blood. Okay, the right atrium pumps blood into your right ventricle, and there's a valve, the right AV valve or the tricuspid valve, between these two chambers. Okay, and then the right ventricle is going to pump that deoxygenated blood to the lungs, so the pulmonary circuit. So you've got through the left and right pulmonary arteries. These arteries are carrying deoxygenated blood. That's why they're color-coded blue. Okay, the lungs oxygenate the blood. So the left side of the heart is receiving oxygenated blood via the pulmonary veins. So that's why these are color-coded red, because the blood is now oxygenated. Okay, the left atrium then pumps blood into the left ventricle, and the left ventricle is going to pump the blood through the systemic circuit, so into the aorta, and that blood is now oxygenated. The two ventricles are separated by what is called the intraventricular septum. Okay, I didn't mention there's also AV valve separating the left atrium from the left ventricle. Okay, so in this class I'm going to call them the right and left AV valve, and the AV stands for atrioventricular valve. Okay, and then there are valves between the ventricles and the arteries. Okay, and these are the semilunar valves, the pulmonary semilunar valve, and then the aortic semilunar valve. Okay, other thing being shown on here is the chordae tendineae. Okay, and the papillary muscles. These are not what open the valves. These are what the valves prevent the valves from opening backwards into the atrium. Okay, and we'll talk about that again. Right, so I mentioned the systemic circuit and the pulmonary circuit. So here we have a more simplified plan for the heart. The right side of the heart receives deoxygenated blood from the systemic circuit. Okay, so all those capillary beds supplying all the tissues that are not your lungs. Okay? Your right ventricle then pumps deoxygenated blood through the pulmonary circuit. And those are all the capillary beds surrounding your lungs, and that's where your blood gets oxygenated. So when it returns to the left atrium, it's fully oxygenated. And then the left ventricle pumps blood through the systemic circuit, where it delivers oxygen to your cells. Okay, the systemic circuit is a series of parallel circuits. So the left side of your heart has to work a lot harder than the right side, because the right side of your heart is only circulating blood through the pulmonary circuit, so through your lungs. And where do your lungs lie in relation to your heart? Really close, right next to it. Okay, so your pulmonary circuit is a low pressure circuit. So the right wall of your right ventricle is really thin. Okay, it doesn't have as much myocardium or muscle to it, because you want low pressure in the pulmonary circuit. And in fact, if you get high pressure in the pulmonary circuit, you push a bunch of fluid into your lungs, and that really impedes respiration. Okay. The systemic circuit has to get blood all the way up to your brain. And where is your brain in relation to your heart? Above it. Okay? And in the blood pressure lab, you're going to take your blood pressure when you're standing versus sitting versus laying. And how do you think your blood pressure differs when you're laying down? Is it higher or lower than when you're standing up? It's lower, right? Your heart doesn't have to work as hard when your head is in line with it. Okay? Your heart is always trying to maintain blood pressure so that your brain is fully vascularized. Okay? Your heart receives blood from the circulatory system outside of it. Oxygen and nutrients. It doesn't receive oxygen and nutrients from the blood inside of the heart. So you have arterioles, capillaries, venules, veins, etc. in the supplying the blood to the heart. Okay, you got the GI tract, and here we have a portal system. Okay, this is the hepatic portal vein that's connecting the GI tract to the liver. Okay, kidneys also have a portal system, so we have the glomeruli and then the peritubular capillaries. Okay, then skeletal muscle is one circuit, skin is another circuit, skeleton, fat, other tissues, etc. So the left side of the heart has to develop a lot more pressure, and therefore the wall of your left ventricle is way thicker. Okay, so your heart receives all the oxygen and nutrients it needs from the coronary circulation, which is in series with the other systemic organs, right? So we have the left ventricles pumping blood into the aorta, and the right coronary artery branches off the aorta. Okay, and then it's going to branch into arterioles and capillaries. Your heart needs a constant supply of oxygen and nutrients because it functions very similarly to your slow oxidative fibers that you find in skeletal muscle. And those slow oxidative fibers have really good endurance, right? And you want your heart to have really good endurance, because it has to contract continuously. Because what happens when your heart stops contracting? You die. Exactly. Okay, hopefully this isn't news to you, right? Your heart is located in your thoracic cavity. Okay, it's about the size of your fist. Okay, your diaphragm is what separates your thoracic cavity from your abdominal cavity. You also have a whole bunch of blood vessels, some nerves, and your lungs occupying your thoracic cavity. Okay? On average, a human heart weighs between 250 and 350 grams. In lab, you guys dissected, were they cow hearts or pig hearts this year? They were pig hearts? Okay, so much larger than a human heart because they're coming from pigs at market weight, which are much larger than the average human. Okay, your heart has three distinct layers. The innermost layer is the endothelium, Endothelial tissue lines your entire vascular tree. Okay, and your heart is no exception. Okay, endothelial tissue is just a type of epithelial tissue that lines the entire vascular tree. The thickest layer is the myocardium. That's the middle layer. That's the muscle. Okay, so the myocardium is the muscle layer of the heart. And then the outer layer is the epicardium, which is just a thin external layer that covers the heart. Right. Other anatomy we need to talk about are the heart valves. The heart valves play a major role in causing unidirectional flow of blood. Okay, it goes from the veins to the atria, from the atria to the ventricles, from the ventricles to the arteries, right? Not the other way around. The heart valves open and close due to pressure differences. Okay, the AV valves, or the atrioventricular valves, open when atrial pressure, the pressure inside the atrium, is greater than the pressure inside the ventricles. Okay, and ventricular pressure is low when the ventricles are relaxing. Okay, so AV valves open when atrial pressure is greater than ventricular pressure. And then they close as soon as ventricular pressure goes above atrial pressure. Right? So they open and close due to pressure changes. Right? So here we're just showing that right AV valve. So when the right atrium, the pressure in the right atrium is greater than the pressure in the right ventricle, AV valve opens. When ventricular pressure increases, AV valve slams shut. And what happens is these papillary muscles, which are branching off of the ventricular wall, when the ventricle contracts, those papillary muscles contract as well. And when they contract, they're pulling on those chordae tendineae because you have all this pressure developing because the ventricles are much thicker than the atria. Okay, so the ventricles develop a load of pressure, and without those chordae tendineae pulling, your AV valves would open backwards and let blood flow back into the atria. Okay? So the papillary muscles contracting and the chordae tendineae aren't opening the AV valves. They're actually preventing the AV valves from opening backwards. Okay, the semilunar valves, right, which are between the left ventricle and the aorta, so that's the aortic valve, and then between the right ventricle and the pulmonary arteries, that's the pulmonary valve. They open when ventricular pressure is above arterial pressure. And they close when arterial pressure is greater than ventricular pressure. Okay. There are no valves between the veins and the atria, right? Because they aren't needed. So atrial pressure is never much greater than venous pressure. So you don't really have to worry about backflow. Plus, when the atria contract, they actually squeeze off the openings to the veins. Okay, so valves aren't necessary between the veins and the atria. Okay, so let's look at those semilunar valves. So here's the aortic valve. When the ventricles are contracting and ventricular pressure goes above aortic pressure, semilunar valve opens. Okay, when the ventricles start to relax, ventricular pressure is going to drop and aortic pressure is not going to drop as much, and so those semilunar valves are going to slam shut. The semilunar valves are under much higher pressure, so when they slam shut, the sound is louder than when the AV valve slams shut. And when you listen for heart sounds, that lub-dub are your valves shutting. So the soft lub is the AV valves, the louder dub is the semilunar valves. Oh, there we go.
The heart is a pump which must circulate blood through two different but interconnected vascular systems. The smaller of these systems is the pulmonary system. Blood returning from the upper part of the body is delivered to the right atrium of the heart by the superior vena cava, one of the body's two largest veins, while blood returning from the lower part of the body is delivered to the right atrium by the other major vein, the inferior vena cava. Contraction of the right atrium in each cardiac cycle forces blood into the right ventricle. This is followed by contraction of the right ventricle, which pumps blood into the pulmonary artery, sending it on through the blood vessels of the lungs. As the right ventricle contracts and pressure within the right ventricle rises, the tricuspid valve situated at the opening between the right atrium and right ventricle shuts, preventing any backflow. The pressure generated by contraction of the right ventricle soon opens the pulmonary valve and blood enters the circulation of the lungs. After passing through the circulation of the lungs, the blood, having been recharged with oxygen and having rid itself of carbon dioxide, is returned through the pulmonary veins to the left atrium. The left atrium, too, contracts, forwarding blood into the left ventricle in order to fill it before it contracts. As the powerful left ventricle contracts, the mitral valve shuts, preventing backflow into the left atrium. The aortic valve opens, and blood is forced into the aorta, which distributes it to the rest of the body apart from the lungs. As the contraction comes to an end and pressure in the aorta falls, the aortic valve snaps shut to prevent backflow into the left ventricle.
Okay, any questions about the heart anatomy? Hopefully it's a review for a bunch of you guys if you've already taken anatomy. Yeah?
I just want to make sure I heard this right. So you said the soft lub sound is the AV valve closing and the louder dub is the semilunar valve.
Exactly, because the semilunar valves slam shut, AV valves close. Yeah. Because the semilunar valves are under much higher pressure than the AV valves. Okay, let's start talking about the electrical activity. So let's start doing some physiology. Okay, so you have a heartbeat, which is a wave of contraction going through the cardiac muscle. Okay, so your atria contract as a unit, right and left, in order to fill the ventricles, and then your ventricles contract as a unit in order to eject blood from the heart. Okay, so this synchronized contraction of atria versus ventricles is highly coordinated. Okay, so your heart propagates action potentials by itself. It is called autorhythmic. Okay, it can propagate action potentials without external input. And it propagates two types of action potentials, right? The autorhythmic cells produce one type of action potential and the contractile cells produce a second. Your autorhythmic cells do not contract themselves. They don't have actin and myosin, right? They're specialized for initiating and conducting action potentials. Okay, so your autorhythmic cells are firing action potentials at a specific rate. And then they're electrically connected to the contractile cells. And 99% of the cardiac muscle cells in your heart are contractile in nature. Okay, they're filled with actin and myosin. When they fire action potentials, they contract as a result. Okay, they do not normally initiate their own action potentials, and you really don't ever want them to. Right? You always want the autorhythmic cells to control those contractile cells so that they contract as a unit. Okay, so here we have an action potential for that autorhythmic cell. Before we talk about it, let's refresh our memories on action potentials in neurons. Okay, so what's resting membrane potential in most of our cells? Negative 70. And what's threshold potential? Negative 55 for muscles, right? And neurons. So skeletal muscle and neurons. Threshold potential is minus 55. Okay, so we have graded potentials that bring us to threshold. What happens at threshold? Yes, I know an action potential happens. So why does an action potential happen?
So because of depolarization.
Depolarization due to sodium rushing in. So voltage-gated sodium channels open at that threshold, right? So we have sodium in, which causes depolarization. What happens at peak, which is at positive 30? What happens at peak? What closes? Voltage-gated sodium closes, and what opens? Voltage-gated potassium opens. So repolarization is due to potassium out. Right? And they stay open a little longer, and so you get after hyperpolarization. And then it returns to rest. So the depolarizing phase of action potentials in neurons and skeletal muscle is due to voltage-gated sodium channels opening, sodium rushing in. And then repolarization is due to opening of voltage-gated potassium channels, potassium rushing out. Okay. In these autorhythmic cells, depolarization is due to calcium rushing in. You look horrified. Okay? So depolarization in the autorhythmic cells is due to calcium influx. And what do you guys know about the amount of calcium versus sodium in extracellular fluid? Is there more sodium or calcium? Way more sodium. Okay, so this is why the peak of depolarization is only just above 0, whereas in an action potential for neurons and skeletal muscles is at positive 30. It's because the equilibrium potential for sodium is much higher than the equilibrium potential for calcium. Okay, so action potentials in these autorhythmic cells peak at a much lower level. Okay. Now, threshold potential is different. It's at minus 40. You might also know that resting is different. It's at minus 60. These autorhythmic cells are weird. And one of the things that's weird about them is they have these channels called funny sodium channels. And they're funny because they're always open, not because they're hilarious. Okay, they're funny weird. Okay, so these funny sodium channels mean that sodium is always coming into the cell, which is making resting membrane potential a little higher than in other cells. Okay, and they never sit at rest. They never sit at this minus 60, because as soon as they hit minus 60, potassium channels start to close, which is also weird. So they have potassium channels that aren't very leaky. They start to close. And then they have funny sodium channels that are always open. So they're always about to depolarize, right? They're trapping in potassium. They're letting sodium in. And then on top of it, they have 2 types of calcium channels, T-type and L-type. Okay? T-type are transient. L-type are long-lasting. Okay? The T-type have a lower threshold than the L-type. So the T-type open during this slow depolarization phase. Okay, and they only stay open for a little bit. But they bring the membrane to threshold, and that threshold is for the L-type calcium. Okay, the L-type calcium channels stay open for longer, and they're what cause that depolarization phase. Okay, at peak, L-type calcium close, voltage-gated potassium open, potassium causes repolarization. So the good news with all these action potentials is that voltage-gated potassium channels always cause repolarization. Okay. These autorhythmic cells never sit at rest. Okay, so that's why there's multiple action potentials being shown. They never sit at rest. Okay, as soon as they hit minus 60, potassium channels start to close, funny sodium are letting sodium in, T-type calcium open, comes to threshold, L-type calcium open, calcium rushes in, depolarization. Okay, voltage gated potassium open, repolarization. Yes, in the back.
Then how do you get variation in heartbeat? If it never rests, would that mean it beats as fast as it possibly can?
No, excellent question. So the autorhythmic cells have an intrinsic firing rate. And then the sympathetic nervous system and parasympathetic nervous system alter that rate, right? So these autorhythmic cells have receptors. They either have muscarinic cholinergic receptors for parasympathetic and adrenergic receptors for sympathetic nervous system. Okay, and so parasympathetic nervous system wants to slow heart rate. So it wants to prevent the depolarization. So it keeps potassium channels open and it keeps calcium channels closed to slow this depolarization. Okay, the sympathetic nervous system wants to speed up the depolarization. And so it opens more sodium channels and it opens those calcium channels, right? So sympathetic and parasympathetic alter that slow depolarization phase. Parasympathetic slows it more, sympathetic speeds it up. Right, so I mentioned they all have an intrinsic rate. So the sinoatrial node or the SA node, which is your pacemaker of your heart, intrinsic firing rate of those action potentials is 80 to 100 action potentials per minute. Is that a good resting heart rate? No. At your age, you should not have a resting heart rate above 80. All right, so what system is slowing your heart rate? Parasympathetic. Exactly. Okay, you also have autorhythmic cells in the AV node. They have a much slower firing rate. Bundle of His and Purkinje fibers. Bundle His runs down the intraventricular septum. Purkinje fibers run up the wall of the ventricles, which again have much slower firing rates. So if your SA gets knocked out, your AV node takes over. Okay, but your heart rate's going to be pretty slow. And if your AV node also gets knocked out, you're in big trouble. Okay, maybe your bundle His will take over, but you're in really big trouble. Okay, we'll talk about the other type of action potential on Friday. But I recommend going back and reviewing neurons to coordinate contraction of the heart, right? We have those autorhythmic cells that spontaneously fire action potentials. Okay? They are what are providing the rhythm to the heartbeat. Okay, and our major pacemaker cells that determine heartbeat are in the SA node, the sinoatrial node, and then in the AV node, the atrioventricular node. Right? But we also have these conduction fibers that transmit action potentials by the pacemaker cells to the myocardium. And the myocardium is made up of the contractile cells. Okay, because we want the atria to contract as a unit before the ventricles contract as a unit. Right? These conduction fibers have a velocity of about 4 meters per second, which is 10 times faster than an ordinary muscle fiber. Okay? And these conduction fibers are especially important in coordinating when the ventricles contract and getting the ventricles to contract as a unit. Because remember, the atria are much smaller. Okay, and the wave of contraction just spreads from cell to cell within the atria. Okay, the SA node fires first, and then it sends a signal to the AV node via the internodal pathway. Okay, so the internodal pathway connects the SA node to the AV node. And then the AV node sends in a signal to the bundle of His, which runs down the intraventricular septum. Okay, and then the Purkinje fibers run up the walls of the ventricles. Because there's way more muscle cells in the ventricles, and so we need these conduction fibers to get them to contract together. Let's look at this in a figure on the next slide, not this one yet. Okay, so this is showing the spread of excitation. Remember, the SA node has the fastest intrinsic firing rate of action potentials. So it fires first. That action potential spreads to the right atrium because the SA node is in the right atrium, which then spreads to the left atrium. This is really rapid. Right and left atria contract as a unit. Okay, push blood into the ventricles. The internodal pathway is one of those conduction fibers that connects the SA node to the AV node. The only electrical connection between the atria and the ventricles is at the AV node. Okay, so the SA node fires, causes the entire atria to contract. Sends a signal to the AV node. There's a delay because you want your atria to contract and relax before your ventricles start to contract in order to be an efficient pump. Okay, so that AV nodal delay is important in allowing the atria to contract fully before the ventricles start to contract. Okay, for ventricular excitation, you want the ventricles to contract from the apex upwards because the arteries are at the top. Okay, so just like with toothpaste, you want to squeeze from the bottom. Okay? You squeeze from the middle. Of course you're a middle squeezer. Okay. So here we have the figure to show you guys that pathway of depolarization. So anytime you see yellow, that means action potentials are being fired. Okay, so the yellow color coding means those cells are firing action potentials. So the SA node has those autorhythmic cells. Okay, they're firing an action potential. It spreads to all the contractile cells in the atria, both in the right atria as well as the left atria. Okay, and then the atria contract as a unit. See, they're all depolarizing at the same time and contracting. Okay, the SA node, via the internodal pathway, causes the AV node to fire an action potential, but there's a slight delay. Okay, so the atria contract, AV node is firing an action potential, and then it sends a signal down the bundle of His, which is in the intraventricular septum, and then up the Purkinje fibers so that the heart contracts from the apex, which is down here, upwards, because here are where the arteries are. Okay, so you want your heart to squeeze from the bottom up, or the apex upwards, just like how you want to squeeze your toothpaste. If you want to get the most for your money. Okay, so atria contract first, followed by the ventricles. The atrial cells, those cells in the SA node, and then all the contractile cells are connected to one another via gap junctions. Right? So all the atrial contractile cells are connected to one another as well as to the SA node via gap junctions. And what are gap junctions? Who remembers? Cell-to-cell adhesion things, yes. You want to elaborate on things?
Well, for neurons, gap junctions involve electrical.
Exactly. So remember we talked about electrical synapses? They involve gap junctions. They're pores between cells, and they allow water-soluble substances to spread from cell to cell. And then in case of excitable cells, those are ions. Okay, so we have what you can think of as being electrical synapses between all those contractile cells in the atria. So they essentially contract as a unit really rapidly. So the action potential spreads from cell to cell really rapidly because of those gap junctions. Okay, and then between the atria and ventricles, there's only a conduction pathway. Then we can get that AV nodal delay. And then we have the conduction pathway that sends the action potential down the septum, up the Purkinje fibers, and then all those ventricular myocardial cells, the contractile cells, are connected to one another via gap junctions. Okay, the cardiomyocytes are connected to one another via intercalated discs. They have both gap junctions and desmosomes. Okay, so you have to think way back to the beginning of the semester where we talked about desmosomes, sort of like spot rivets. Okay, and then we talked about gap junctions, but then we talked about them with electrical synapses as well. Okay, so here we have one of those autorhythmic cells in the SA node. Okay, it does not contract. It just has those funny sodium channels, the T-type and L-type calcium channels that cause it to spontaneously fire action potentials. Right, but it is electrically connected via gap junctions to the cardiomyocytes that do contract, the contractile ones. So here we have a blow-up at the junction between two adjacent cardiomyocytes or contractile cells. And I know they're contractile cells because they have sarcomeres, right, which are made up of thick and thin filaments, which should look familiar to you because we talked about sarcomeres when we talked about skeletal muscle, right? What is different between cardiomyocytes and skeletal muscle is the fact that those adjacent muscle fibers are connected to one another via gap junctions and desmosomes. And together, the desmosomes and gap junctions are called intercalated discs. Okay? Every time your heart fills with blood, it stretches, right? And you don't want the blood to leak out between the cells, right? So hence, we've got desmosomes to keep those two cells adjacent to one another so that blood's not leaking in between them. Okay. So let's compare and contrast cardiac muscle to skeletal muscle. Since we just spent a whole week talking about skeletal muscle. Both cardiac muscle and skeletal muscle are striated. And those striations come from sarcomeres. So the arrangement of thick and thin filaments. Okay, so both skeletal muscle and cardiac muscle have sarcomeres. Okay, both skeletal muscle and cardiac muscle have troponin and tropomyosin to regulate contraction. What does troponin do? Which one covers the binding sites for myosin on actin? Tropomyosin. Which one binds calcium and moves tropomyosin out of the way? Troponin. Okay? So both skeletal muscle and cardiac muscle use troponin and tropomyosin. There's a troponin C molecule that they test your blood for if they think you've had a heart attack. Okay, so that's the cardiac form of troponin. So if you are releasing a whole bunch of troponin C into your bloodstream, that means there's been some sort of cardiac event. Okay, that your heart muscle has been damaged. Okay, both skeletal muscle and cardiac muscle have T-tubules. What are T-tubules?
They allow the potential to travel down.
They allow the action potential to travel down into the interior of the cell. They're invaginations of the sarcolemma, the plasma membrane. Okay, cardiac muscle also has sarcoplasmic reticulum, but it's not quite as well developed as it is in skeletal muscle. And what does sarcoplasmic reticulum do for skeletal muscle?
Holds calcium.
It holds calcium. Okay? And the reason why cardiac muscle doesn't have as much sarcoplasmic reticulum is because it lets calcium in from extracellular fluid as well. Okay, so it doesn't need as much intracellular storage of calcium because it also gets some of the calcium for contraction from extracellular sources. We'll talk about that in a minute. Okay, our cardiac muscle is very similar to our slow oxidative fibers. So if any of you have ever eaten heart, okay, so if you eat beef heart, it's super lean. So a lot of people don't eat it because it's kind of chewy, right? But my guess is some of you have had chicken hearts, especially if you go to those Brazilian barbecue places where they come around on skewers. Okay, it's always really dark red in appearance. And what causes it to be dark red in appearance? Which one of those molecules listed gives it that dark red color? Myoglobin. And what's myoglobin? What's myoglobin? What does myoglobin do for muscle? It's an oxygen carrier that's within muscle. Okay, so these are oxidative fibers. They have oxygen within them being held by the myoglobin. They have loads of mitochondria, in order to do aerobic respiration. Okay, so they are not producing lactic acid. So they are super slow to fatigue, which is really good news, because what happens if your heart fatigues? You die. Yep. Okay, now let's talk about differences between cardiac muscle and skeletal muscle. Cardiac muscle has those intercalated discs. Okay, so there's desmosomes and gap junctions connecting adjacent muscle fibers. Okay, that does not occur in skeletal muscle. Okay, cardiac muscle is innervated by the autonomic nervous system. Which branch of the peripheral nervous system innervates your skeletal muscle? Somatic, exactly. Okay. Your cardiac muscle is influenced by hormones, in specific norepinephrine and epinephrine, greatly influence heart rate. Okay. And then, I already mentioned this, calcium that regulates contraction comes from both extracellular fluid and the sarcoplasmic reticulum. Okay. Cardiac contractile muscle cells also fire different action potentials than skeletal muscles. Right? So cardiac contractile muscles fire different action potentials than skeletal muscles. So we have our neuron and skeletal muscle action potential up on the board. We've got our autorhythmic cardiac cell action potential up on the board. And now let's add the cardiac contractile muscle cell action potential. So here we have an even lower threshold potential. So you have resting potential at minus 90 millivolts. Okay, and then we're going to set threshold potential at minus 70 millivolts, right? And then we're going to draw depolarization with the peak at positive 30 millivolts. Okay, so sits at a lower resting membrane potential, and the threshold potential for the voltage-gated sodium channels is a little lower in these contractile cells in the heart. Okay, so cardiomyocytes have a lower resting membrane potential and a slightly lower threshold potential for their voltage-gated sodium channels.
The image makes it look like it's a little above plus 30. Is it supposed to be at plus 30 though?
Yeah, it's at about plus 30.
Okay.
Yeah. Yeah. And depending what textbook you look at, it's not always plus 30 anyway. It's just right around plus 30.
This class is plus 30.
Yep, for this class it's plus 30. Okay, so depolarization phase of the contractile cardiomyocytes is just like in skeletal muscle. It's due to voltage-gated sodium channels opening, sodium rushes in. Okay, so that's the same. At peak, those voltage-gated sodium channels close, and then L-type calcium channels open. So that is the big difference. Okay, the contractile cardiac cells express L-type calcium channels. Okay, when those L-type calcium channels open, calcium is able to rush in, but because the equilibrium potential for calcium is lower than sodium, that's why you get a drop. Right, so we get this drop in membrane potential because calcium's equilibrium potential is lower than sodium's. Okay, and then that is what causes the plateau phase. Right, so it's L-type calcium channels open, leading to a plateau phase. So you get this extended depolarization. Right, those calcium channels then close, and potassium channels open, and so repolarization is always due to potassium out. Okay, so we've got depolarization due to voltage-gated sodium channels. Sodium rushes in. At peak, sodium channels close. So this is why we get a decrease in the permeability to sodium. Okay, L-type calcium channels open. That's why increase in permeability to calcium. And that's what the L is for, the long-lasting or L-type. That leads to the plateau phase. Calcium channels close, which decreases the permeability to calcium. Voltage-gated potassium open, potassium rushes out. Okay, so we get this plateau phase, extended phase of depolarization. Okay, and I mentioned that calcium comes from both the sarcoplasmic reticulum and from extracellular fluid. Okay, so those L-type calcium channels are letting calcium in from extracellular fluid. And troponin doesn't care. Okay, it just binds to calcium. It doesn't care if it's from intracellular stores or extracellular fluid.
I have a question.
Oh, yeah.
So calcium is positively charged. Why does that cause a drop in the membrane potential?
Okay, it causes a drop in the membrane potential because the equilibrium potential for calcium is lower. So the sodium-potassium pumps are running this whole time. So sodium starts getting kicked out, and only so much calcium is available to come in to maintain that depolarization.
Okay, so there's more sodium leaving than the calcium.
Exactly, right? So sodium's getting pumped out immediately, and there's only so much calcium to replace it. Okay. Yeah.
Is there a reason why this potential is so much bigger? Like, does it result in a stronger contraction?
We're going to talk about that right now. Yes, that plateau phase is super important. Okay, it is super important because it prevents summation and tetanus. Okay, the plateau phase of the action potential in the contractile cardiac cells prevents summation and tetanus. Why don't you want your heart to do tetanus? Because you die. Yeah. So you would squeeze, and then you would squeeze a little harder, and then you would squeeze a little harder. That's not a good pump, right? You want your heart to contract, and then you want your heart to relax. And then you want your heart to contract, and then you want it to relax. Okay? And without that plateau phase, you could potentially get summation. So it would contract and then contract a little harder. And then it might relax, right? Or you could get tetanus, where it would contract, and then it would contract, and then it would contract even harder, right? And it would stay contracted, and you're not going to fill the ventricles. You're going to do one last ejection, and that would be it. Okay? So here we have membrane potential, which goes with the red line, and relative cardiac muscle fiber tension, which goes with the blue line. Okay, so our cardiac muscles contract and then they relax because there is an extended absolute refractory period. Okay, there's a greatly extended absolute refractory period during the plateau phase, which prevents summation and tetanus. Right. So here we have a nice cartoon showing excitation contraction coupling in those contractile cells. Okay, so they're connected to one another via gap junctions. So adjacent cardiomyocytes depolarize their neighbor. Right? So the action potential just flows from cell to cell. Right? Once the cell depolarizes, right, those L-type calcium channels are going to open. So this is the plateau phase. Okay, so the L-type calcium channels open, right, as well as the action potential going down the T-tubules are going to allow those RYR receptors to open and allow calcium to flow out of the sarcoplasmic reticulum. Okay, so the L-type calcium channels causing the plateau phase, and then the depolarization going down the T-tubules, open those RYR channels, and allows calcium out of the sarcoplasmic reticulum. All that calcium is going to bind to troponin. Troponin will move tropomyosin off the binding sites on actin for myosin, those little black dots, which will allow cross-bridge cycling. Cross-bridge cycling is identical. Okay, so we're not going to go over it again. For relaxation to occur, calcium has to be removed from the sarcoplasm. Just like in skeletal muscle, it gets pumped back into the sarcoplasmic reticulum. okay, by a calcium ATPase. Uses one molecule ATP to pump one calcium back into the sarcoplasmic reticulum. Okay, you want relaxation to occur fully before the next round of contraction. So calcium also gets pumped out into the extracellular fluid by another calcium ATPase on the sarcolemma, as well as a calcium sodium exchanger. So this would be a secondary active transporters. These are primary active transporters. Okay, so 3 methods to pump calcium out of the sarcoplasm to get full relaxation before the next round of contraction. Okay, so removal of calcium in cardiac muscle involves a calcium ATPase and sarcoplasmic reticulum. This is what muscles do, skeletal muscles do. Okay, in addition, the cardiac muscles do calcium ATPase on the plasma membrane or sarcolemma, right? And then a sodium calcium exchanger on the sarcolemma. Okay, these two are primary active transport. This one's secondary active transport. Once calcium gets removed, troponin moves tropomyosin back over the binding sites, cross-bridge cycling stops, ventricles totally relax and fill with blood. Okay, so there is so much electrical activity that gets coordinated in your heart, it can get picked up on your skin surface in the form of an ECG, an electrocardiogram. Right? And did you guys do those in lab this week? Yeah, with just the 3 lead. Okay, when you have an actual ECG at the doctor's office, or hopefully not in the ambulance, right? It'll be a 12 lead one, which will be a lot better. Okay, so what's happening down here, we have an ECG. So this is what shows up on the trace paper or on the computer screen if you're using BioPack, right? Okay, so we've got the P QRS complex and then the T wave. So the P wave, QRS complex, and the T wave. And then we've got these different intervals. So we've got the PQ segments, right? The R to R interval, the T to Q segment, and the Q to T interval. And whether it's a segment or an interval, don't ask me why they named one segments and one intervals, okay? What's up here is the membrane potential in the ventricles. Okay, so it's showing the membrane potential in the ventricles in specific. Okay, during the P wave, your atria are depolarizing. During the QRS complex, your ventricles are depolarizing. Which one has more electrical activity based on the ECG? Ventricles. Why do the ventricles have more electrical activity? They're bigger, exactly. Okay. Right. The QRS complex, the depolarization of the ventricles, hides atrial repolarization. Okay, so atrial repolarization occurs in the QRS complex. And then the T wave is showing ventricular repolarization. Okay, the difference between P and Q is showing you that AV nodal delay. Right, so that the atria contract as a unit, and then the ventricles wait until the atria are done. Okay, from the Q to the QRS complex to the T wave is what's called ventricular systole. So that's when the ventricles are contracting. So systole means contraction. Notice your ventricles relax a lot more than they contract. Okay? And this allows them to not undergo fatigue. They spend a lot longer relaxing than contracting. Okay, so the T to Q interval is ventricular diastole, or when the ventricles are relaxing. Okay, so on quizzes and exams, I'll ask you what the P wave represents, okay, or what the T wave represents, or what the segment represents, okay? And we'll ask you to diagnose a condition based on the ECG. Yeah?
So the atria are contracting during the QRS complex.
No, they're relaxing.
They're relaxing, so are they contracting during the part of the P wave?
So they're contracting at the start of the P wave, and by the time you have R, they've relaxed. So the atria contract for an even shorter period of time.
Okay, so between P and R, they've contracted and relaxed.
Mm-hmm.
And then the ventricles are contracting between the QT.
Right. Q to T is ventricular contraction. You can think P to Q is atrial contraction.
Okay.
Yeah. Okay. So your heart spends a lot of time relaxing, which is good news.
So
You want it well rested.
When they start contracting, is that at the top of the waves? You know, once they've completely depolarized, is that when contraction starts, or does contraction start during depolarization?
It starts during depolarization. Yep. Okay, so it would start at the beginning of the P wave. You'd start seeing contraction in the atria.
Okay, and then you'd start seeing contraction in the ventricle
At the
between
start,
P and R.
yep, at the start of the Q wave.
Okay.
QRS complex, not the Q wave. Okay. Right. So we're going to put all this stuff together and talk about the cardiac cycle. We're going to put the electrical activity together with pressure changes, with opening and closing of valves. Okay, so the cardiac cycle is electrical activity as the heart depolarizes and repolarizes due to that autorhythmic cells. Okay, specifically the autorhythmic cells in the SA node, which are the pacemaker cells. Okay, and then it's the mechanical activity, so the contracting and relaxing. So systole means contraction, diastole means relaxation. And when we say the heart is in systole, we mean the ventricles are in systole. Okay, but the atria undergo systole as well. It's just that they're really puny, so we don't think of them, you know, when we say systole, we're meaning the ventricles. Okay, and then the alternate contraction and relaxation of the atria versus the ventricles determines pressure changes, which determines directional flow. Okay, because remember, those heart valves open due to pressure differences. And those pressure differences are due to contraction versus relaxation. Okay, so we're going to put all three of these events together to talk about the cardiac cycle. Okay, we're going to separate the cardiac cycle into 4 phases. And they're descriptive. So the first phase, because you just have to start somewhere, is ventricular filling. So based on that description, what's happening? Ventricles are filling with blood. Okay? The second phase is the first isovolumetric phase. Okay, so it's isovolumetric ventricular contraction. Okay, so what does iso mean? Equal. So isovolumetric means no change in volume. So that means blood's not entering or leaving the ventricles. Okay, and the ventricles are contracting. So as long as you know what isovolumetric means, this is really easy. So no change in volumes, but the ventricle is contracting. Okay, third phase is ventricular ejection. And it's on the next slide. Okay, we're just going to put them all up on the board for now. So you ventricular ejection, what do you think is happening to the blood? It's leaving the ventricle. Okay, and then the last phase is isovolumetric ventricular relaxation. Okay, so isovolumetric, no change in volume. This time the ventricles are relaxing though. Okay, in order for the ventricles to fill with blood, what valves have to be open?
Atrioventricular valve.
Yep, the AV valves have to be open. Okay, are any valves open if you have no change in volume? No, so no valves open. Okay, in order for blood to leave the heart, what valves have to be open? Semilunar. Okay, and then no valves open if no blood's moving. notice there's never a phase when all the valves are open. Never choose that. That is never the answer. Okay, so if I put a distractor and it says all 4 valves are open, you know it's not correct. Guess something else. Okay. It's just like it's never action potential magnitude changes. Okay. Though people still always choose it. Right? So, valves open and close due to pressure changes or pressure differences. For the AV valves to be open, atrial pressure has to be above ventricular pressure. Okay, so atrial, so that's the pressure in the atria. Atrial pressure, I'm just going to say P, okay, is greater than ventricular pressure. Okay, and remember Pac-Man eats the bigger number. It's not the alligator. I grew up in Pac-Man time. It's Pac-Man, okay? For no valves to be open, right, this means that ventricular pressure must be above atrial pressure now. Right? And let's talk about getting those semilunar valves open before we fill out the rest of these pressure changes. So semilunar valves open. When are the semilunar valves open?
The pressure on the ventricle exceeds that of the arteries.
Exactly. So this is when ventricular pressure is greater than arterial pressure. Okay, so make sure you know the difference between atrial pressure and arterial pressure. Atrial pressure is the pressure in the atria. Arterial pressure is the pressure in the arteries. Okay, so when ventricular pressure is greater than arterial pressure, those semilunar valves will open. Okay, so if the semilunars valves are closed, as they are in all other stages except for ventricular ejection, that means that ventricular pressure is less than arterial. Right, and that's the same here. And the same here. Okay. And for this isovolumetric stage, that means that ventricular pressure is also lower than atrial. Okay, to get those AV valves closed. Right, in ventricular filling, it can be subdivided into 2 phases. Yeah.
So is it atrial or arterial on isometric ventricular relaxation? Because if they're higher, because you have it written that ventricular pressures are lower than atrial, shouldn't it be filling at that point?
Oh, God, you're right. I made a mistake, you guys. You're right. Ventricular pressure is still above atrial pressure.
But less than arterial.
Less than arterial. Thank you. Yes. Okay. Ventricular pressure is greater than atrial pressure. That keeps the AV valves closed. Okay. And then arterial pressure is greater than ventricular pressure, and that is closing the semilunar valves. And that's the same for both isovolumetric phases. Yes. Okay. Back to ventricular filling. There's 2 phases separated. There's the passive phase and the active phase. During the passive phase, the atria and the ventricles aren't contracting. But the reason atrial pressure is above ventricular pressure is that blood is returning to the heart. So it's actually venous pressure that's above ventricular pressure. And that's what's filling the ventricles. During the active phase, the atria are actually contracting and squeezing a little bit more blood into the ventricles. Okay, during the isovolumetric phases, no valves are open, no movement of blood. However, at this point, ventricular myocardium is starting to contract. It just hasn't contracted hard enough yet to get above arterial pressure. Okay, once it gets hard enough to get above arterial pressure, we enter ventricular ejection, semilunar valves open, right? Blood exits the ventricles. As soon as those ventricles start to relax, right, ventricular pressure drops below arterial pressure. Those semilunar valves close. AVs are still closed. Again, no movement of blood. Right? Let's look at that in a series of figures. So in your textbook, this is all lined up on one page, right? But I've split it into multiple slides because it's too long of a figure for one slide. So here we have the heart, and we are showing what's happening during the various phases. So we've got ventricular filling. This is the passive phase. The atria aren't contracting yet. Notice we have ventricular filling over here as well. Okay, so it's a cycle, right? So the ventricles start to fill, and this is just due to venous pressure from blood returning to the heart. At the end of ventricular filling, the atria actually contract. Okay, AV valves are open in order for the ventricles to fill with blood. Semilunar are closed. Okay. Phase 2 is isovolumetric ventricular contraction. So as soon as the ventricles start to contract, those AV valves slam shut. Notice no valves are open. Okay, this is a pretty short phase. Phase 3 is ventricular ejection. The ventricles have contracted hard enough that the semilunar valves are open. Okay, so no semilunar valves are open. Blood is exiting the heart. And then we enter phase 4. As soon as the ventricles start to relax, semilunar valves slam shut, no valves are open. It's an isovolumetric phase. Again, this is a pretty short phase. Notice phase 1 is the longest phase. Ventricular filling is the longest phase. Okay. Starts over here, continues again because it's a cycle. Okay, so here we just have the phases listed. We're not showing blood in the heart anymore. We've got which valves are open versus closed. And here we have actual pressure readings. And we have atrial pressure being shown in pink and ventricular pressure shown in green. Notice that atrial pressure never gets that high. The atria are puny. Okay, they can't develop a lot of pressure. All they have to do is get 1 millimeter mercury above ventricular pressure, though, to open those AV valves. So notice the pink line is just above the green one. Whenever it's above the green one, AV valves are open. Whenever the AV valves are open, you're in ventricular filling. Okay, as soon as the ventricles start to contract, those AV valves are going to slam shut, and those ventricles can really develop pressure. Okay, they're going all the way up to 120, which is way above atrial pressure. So of course the AV valves are closed, and the ventricles have to relax. And as soon as they enter a pressure reading below the atria, those AV valves open again. But again, notice the atria are never much higher in pressure than the ventricles during ventricular filling. They just don't develop a ton of pressure. Okay, this would be the active phase right here, when they're actually contracting. The rest of the time, all this pressure is just due to blood returning to the heart, so it's venous pressure. Okay, now we have pressure differences between arteries and ventricles. So green line again is ventricular pressure, and then the blue line is aortic pressure. So we're going to just show the aorta, because the pulmonary artery actually has much lower pressure than the aorta. Okay, so the aortic pressure, notice it's just going between 80 and 120. So you guys probably know average blood pressure for humans is 120 over 80. 120 is systolic. That's the pressure when the ventricles are contracting. And the other is diastolic, the 80, so the DP is diastolic pressure. That's when the ventricles are relaxing. Okay, so notice aortic pressure is most of the time much above ventricular pressure. Okay, so your major arteries serve as your pressure reservoirs. It allows your heart to fully relax so that blood will return to it. Because to be an efficient pump, your heart has to be the lowest pressure point at times and the highest pressure point in the circuit. Okay, so your heart has to fully relax, but your aortas maintain a forward flowing pressure, right? They never go much below 80. And then this line here is your mean arterial pressure. So the pressure on the rest of the circuit. Okay, as the ventricles start to contract, They just have to go slightly above aortic pressure in order for those semilunar valves to open. As soon as they open, you're in ventricular ejection. Okay, as soon as they start to relax, aortic pressure is going to go above ventricular pressure because your arteries are really elastic. And as soon as those semilunar valves slam shut, you get this little upward blip in blood pressure, which is called the dicrotic notch. That's due to the recoil of your arteries. So they stretch during ventricular ejection, and then once blood is no longer flowing from the ventricles, they recoil, right? And that recoil gives you this little upward blip or dicrotic notch. And then pressure slowly falls as blood drains off through the vascular tree, okay, until the next round of ventricular ejection. Hey, this figure is showing volume of blood in the ventricles. So obviously it's increasing during ventricular filling. Okay? And then it stays the same in the isovolumetric phase. So this phase 2 is isovolumetric, no change in volume. Then it decreases during ventricular ejection. It stays the same during the isovolumetric phase, and then it starts to increase again during ventricular filling. Okay, and you get a little faster filling when the atria actually contract. What this is showing is names of some volumes. End diastolic volume is the maximum amount of blood that's in the ventricles at the end of their diastole, their relaxation period. Okay, so the maximum amount of blood in the ventricles. End systolic volume is the minimum amount of blood left in the ventricles at the end of systole, so at the end of ventricular ejection. Okay, so end diastolic, maximum blood at the end of ventricular filling. End systolic, minimum blood at the end of ventricular ejection. End diastolic minus end systolic is stroke volume. That's the amount of blood pumped out of your heart with each heartbeat. Okay, notice end systolic volume is not 0. Right? There's always some blood that remains in your heart, even though it's squeezing the most efficient way from the base upward. Okay. Last thing with cardiac cycle is the heart sounds. So when you're listening with a stethoscope, you're hearing those heart valves closing. Okay, the first sound, the soft lub, is when the AV valves close. So here's the lub, which is at the end of ventricular filling. The louder dub occurs when the semilunar valves close because they're under much higher pressure, and that occurs at the end of ventricular ejection. Okay, so soft lub is the end of ventricular filling. Loud dub is the end of ventricular ejection. Right, and again, your book stacks these all together. Okay, so we're going to finish cardiac physiology by talking about control of cardiac output. Right. And first, you need to know what cardiac output is. So cardiac output is heart rate times stroke volume. Okay, so cardiac output is heart rate times stroke volume. And if you remember way back before spring break, okay, stroke volume is equal to the maximum amount of blood in the ventricles, which is end diastolic volume. So the amount of blood in the ventricles at the end of diastole, which is the end of ventricular filling. Okay, minus the minimum amount of blood ever in the ventricles, which is at the end of systole, and that's end systolic volume. So that's at the end of ventricular ejection. Okay, so stroke volume is the volume of blood pumped out of the ventricles each time. Okay, heart rate is the major determinant of cardiac output because you can ramp up and ramp down heart rate to a greater extent than you can change stroke volume. Okay, heart rate is more variable than stroke volume. Okay. Parasympathetic nervous system slows your heart rate. Thank you. Sympathetic speeds it up, which we already talked about when we talked about the peripheral nervous system. On average, an average cardiac output, if you have a heart rate of 70 beats per minute and an average stroke volume of 70 mils per beat, that's 4.9 liters of blood per minute. Okay, so those of you who have already been sitting down for a few minutes and your heart rate has come back down after that long walk up the one flight of stairs into the lecture hall, okay, or running from your car, you're pumping around 5 liters of blood per minute from your heart. Okay, when you start to exercise, if you're nice and young, you can get your heart rate up to 300. Okay, but as you age, your max heart rate is much lower than 300. Okay, and then notice stroke volume only increases to 100. Okay, so much less variable. So you can go from pumping around 5 liters of blood to pumping 30 liters of blood per minute when exercising. Okay, heart rate is the major determinant. Question?
So how do you increase stroke volume? Because I know that enlarged heart is actually a very bad thing.
Mm-hmm. Yeah, so stroke volume is increased by increasing end diastolic volume, and you can increase end diastolic volume by increasing venous return. And the way you increase venous return is by having your veins vasoconstrict, right? And when they vasoconstrict, it's usually because the sympathetic nervous system tells them to. Okay, and then you can also increase stroke volume by decreasing end systolic volume, right? And you decrease end systolic volume by squeezing harder, and the sympathetic nervous system tells your ventricles to squeeze harder. Okay, so the sympathetic nervous system also controls stroke volume. Okay, it increases cardiac output by increasing overall heart rate, but then it also increases stroke volume by causing more venous return, so better filling of the heart, and then causes your heart to squeeze harder. Okay, so here we have the autonomic inputs to the heart. Remember, the autonomic nervous system is a 2-neuron chain. The sympathetic cardiac nerve comes from the preganglionic neurons from T1 through T4. Notice that the sympathetic postganglionic neurons that are part of the cardiac nerve innervate the SA node, okay, innervate the AV node, and innervate ventricular myocardium. Whereas parasympathetic nervous system via the vagus nerve only innervates the SA node, and here we've got the postganglionic neuron right here. Okay, ganglion's really close to the effector. Here's the postganglionic neuron innervating the AV node. Okay, because when you speed up the SA node, you also want to speed up the AV node. When you slow down the SA node, you also want to slow down the AV node. So that's why both sympathetic and parasympathetic innervate both the SA and AV nodes. Only the sympathetic nervous system innervates the ventricular myocardium, and that's what tells it to squeeze harder. Okay. Right. Your SA node has an intrinsic firing rate due to those autorhythmic cells that have the funny sodium channels and the T-type and L-type calcium channels. Hopefully some of this is familiar after spring break. Okay, has an intrinsic firing rate of about 100 beats per minute. Is that a good resting heart rate? No, that is not a good resting heart rate. Okay, so your SA node is under the control of the autonomic nervous system, so sympathetic and parasympathetic, as well as hormones. During rest, your parasympathetic nervous system slows your heart rate, and here it's saying an average heart rate of 75. That's a little high, especially for young people, okay, closer to between 60 and 70 is where your resting heart rate should be. Okay, and then excitement, sympathetic takes over and heart rate increases. So what we have down here is membrane potential versus time. Okay, and the blue line is parasympathetic frequency. Notice that there's only one action potential. Okay, whereas at rest, we've got the gray line, we've got 2 occurring in the 1.4 seconds. And with sympathetic stimulation, it increases the firing rate. And we've got 3 action potentials in 1.4 seconds. Okay, so we're going to talk about the mechanism of how parasympathetic and sympathetic nervous system increase and decrease heart rate. This is a good review of how those autorhythmic cells fire action potentials. Okay, so sympathetic nervous system increases heart rate. And what we're going to do is talk about the exact mechanism it increases heart rate. So your SA node has beta-1 adrenergic receptors, which bind to both epinephrine and norepinephrine. Okay, so when norepinephrine gets released by the sympathetic postganglionic neuron, it's going to bind to those beta-1 adrenergic receptors. And then when epinephrine gets released by your adrenal medulla, it will also bind to those beta-1 adrenergic receptors. Okay, so it's under direct innervation control by the sympathetic as well as hormonal control from the adrenal medulla. When epinephrine or norepinephrine binds to those adrenergic receptors, it increases the open state of those funny sodium channels, right? And they're funny because they're always open. Okay? If more sodium channels open, you're going to get faster depolarization. Okay? It also increases the open state of the T-type calcium channels. And again, calcium is going to rush in and depolarize the cell. That increases spontaneous depolarization rate, increases heart rate. So let's look at this with a little cartoon. So here we have the plasma membrane of one of those autorhythmic cells in the SA node. They have funny sodium channels and they have the T-type calcium channels. Not shown as the L-type calcium channels, right? But they also have those L-type calcium channels as well. Here's the beta-1 adrenergic receptor. So what happens is when norepinephrine or epinephrine binds, it activates a G protein. So this blue thing on the interior portion of the plasma membrane is what's called a G protein. Okay, when the beta-1 adrenergic receptor binds to norepinephrine or epinephrine, the G protein gets activated. It comes over and it starts the adenylate cyclase. And that adenylate cyclase starts to break down ATP into what's called cyclic AMP. And this is the second messenger. Okay, this is the first messenger. Norepinephrine and epinephrine are the first messenger. Cyclic AMP is the second messenger. Okay, and the process of activating or making cyclic AMP, this whole process is called signal transduction. Okay, so epinephrine and norepinephrine never enter the cell. They bind to a receptor and the signal gets transduced to a second messenger, which is cyclic AMP. Cyclic AMP then activates these protein kinases, and what the protein kinases do is they phosphorylate more funny sodium channels and T-type calcium channels, which opens them. Right? And if you open more sodium channels and calcium channels, sodium and calcium enter the cell and it depolarizes. Right. Let's talk about parasympathetic. Okay, so increased parasympathetic activity. So when the vagus nerve is firing, that postganglionic neuron is going to release acetylcholine as its neurotransmitter. The SA node also expresses muscarinic cholinergic receptors, which bind to the acetylcholine. And when acetylcholine binds to the muscarinic cholinergic receptors, that increases the open state of potassium channels. So remember, one of the things that happens is potassium channels close during that pacemaker potential in those autorhythmic cells. Keeps potassium channels open, potassium continues to leave. Right? And then it keeps calcium channels closed so that calcium can't enter. This is going to decrease depolarization rate, slow the heart rate. Okay, and let's look at this in the cartoon as well. So here we have the plasma membrane again of one of the cells in your SA node, and here it's expressing the muscarinic cholinergic receptor, which is also G protein coupled. When acetylcholine binds, activates these G proteins, and this is a way simpler cascade of reactions. Those G proteins just either keep potassium channels open or T-type calcium channels closed. Okay, no second messenger involved. Those G proteins get activated, keep potassium channels open so potassium can leave, keep calcium channels closed so that calcium can't enter. Okay, so a really nice refresher about sympathetic and parasympathetic nervous system. So I get to ask you questions about that as well on exam 3. Okay, so stroke volume can also be adjusted. So that's what's being shown in this flow diagram. Okay, but I also went over this with the question earlier. Okay, so an increase in venous return is going to increase end diastolic volume. When you increase end diastolic volume, you increase stroke volume. An increased venous return occurs due to venous vasoconstriction. So when your veins vasoconstrict, there's only one way for the blood to go, and that's back to the heart. Okay, because veins have valves, one-way valves. Okay. Increased sympathetic activity or epinephrine directly to the ventricular myocardium increases contraction of the ventricles. And when they squeeze harder, they squeeze out more blood and they decrease end systolic volume. Okay, so increasing end diastolic volume and decreasing end systolic volume both increase stroke volume. Okay, the other thing that can increase stroke volume is a decrease in arterial pressure. Okay, so if there's a decrease in arterial pressure, your ventricles don't have to work as hard to squeeze blood out. This happens when you have vasodilation occur of arterioles supplying skeletal muscle. Okay, so when you turn on the spigots to your skeletal muscles, that decreases arterial pressure and your heart doesn't actually have to work as hard. Okay, in order to squeeze out a little extra blood. Okay, so let's put it all together for cardiac output. Okay, remember cardiac output is heart rate times stroke volume. An increase in sympathetic activity to the heart both increases stroke volume and heart rate, which increases cardiac output. Okay, in order to decrease cardiac output, parasympathetic nervous system can slow your heart rate, right? And that can help decrease cardiac output. But parasympathetic nervous system doesn't have any effect on stroke volume. Okay. So we can ramp up cardiac output really fast, but it takes a little time in order to decrease cardiac output. Right. Any questions about cardiac physiology before we move to talk about blood vessels?